Hello students, how are you? How was your preparation going on? Shanta, how was your preparation going on, ma? Nandish? Okay, so guys, before going to start the class, so you know that, right? Today we are going to talk about the principle of inheritance and the variation and also the molecular basis of inheritance. Okay, so from this chapter, each chapter you will get the four questions. Okay, minimum four questions you will get. So be prepared for this chapter. This is a very important chapter. Okay, fine. So let us revise this chapter, guys. Good morning, Shanta. Are you ready, guys? Tell me whether I'm audible and screen is visible. YouTube people. Good morning, all. How was the Josh, guys? I'm not getting any type of Josh from you. Tell me, guys, how was the Josh? You're preparing for the exam. And I always tell you, don't fear about the exams. Okay? Celebrate the exams as a festival, guys. Because you read for the 2 years, right? Celebrate your exams as a festival. Be happy when you're writing the exams. Okay, fine. So let us start the class, guys. Okay, fine. Let us start the class. Okay. So I want to know that. So what is the genetics actually? Can you tell me, guys, what is genetics actually? I need answer from you because these are your chapters, right? Tell me what is genetics means, guys. Can anyone tell me? I need answer from you. This is a revision. Tell me, what do you mean by genetics? You all know that genetics is a branch of biology that is deals with heredity and the genetic variation. Right, guys? Heredity and the genetic variation. For example, if you take how the character was passing from the parent to the offsprings, right? So that is we are going to study. And some offsprings are differ from the parents, right? So there is a variation was happened, right? So those all the things we are going to study in the genetics, right? Yes. So you know that. So when the genetics was started, on the time we don't know what is a gene, right? So our mental used it as a factor. Some factor is going to be transferred from the parent to the offspring. So he used it as a factor, right? On the time we don't know what is gene and what is the DNA structure and all the things, right, guys? Yes. So now you tell me who is the father of genetics, guys? Who is the father of genetics? I need answer from you. Who is the father of genetics? Study of the traits. Yes, Mendel, right? Yes. The father of the genetics was Gregor Mendel. Okay, fine. So let us come. So what is the genetics, guys? It is the study of the heredity and the genetic variation. And what do you mean by heredity? You all know that the characters which is passing from parental generation, not the character, trait. Okay, not the character. It is a trait. Some of them will confuse between the trait and character. Let us study about that. Okay. So the transfer of trait from the parental to the offspring, we will call it as a heredity. Okay. So what do you mean by character and what do you mean by trait? I will tell you. So many of them will get the confusion about what is the trait and what is the character. I will tell you, guys. Character means what? For example, if you take a eye color. Okay. So eye color is a character. Okay. My eye color is brown. Okay. And maybe your eye color is the black color or the blue color. Okay. So those things we'll call it as a trait. Okay. My brown color of the eye is due to the I have because the trait was transferred from my parent, right? And your black color of the eye, that is also due to your parent, right? So that is what we will call as a trait. Here, character is not transforming, okay? Where trait is going to be transferred, passed from the parent to the offspring. Okay, guys, remember, eye color is a character, whereas the trait is brown, black, and the blue color. Okay, and hair color is the character. Either it may be a blonde hair, some person will not have the hair blonde hair, or black color, or the brown color. That is the difference. Okay, and coming to the height of the person, some are having the height, some are having very short, that is the dwarf, and some are having the intermediate height, right? So those all are the trait. Okay, based on the parents, it will be depend, right? Yes. So then coming to the what is the variation? All of you know, all of you study the evolution chapter also, right? Variation is very important in the evolution, right or not, guys? Tell me, right or not? Yes, Gregor John Mendel. Yes, Nandish. Tell me. So variation is important for the evolution or not? Tell me, without the variation, evolution is happened or not, guys? Tell me. Without the variation, evolution was happen, will not? Yes. Variation will lead to the evolution. Right, guys? Yes. So we have to do the variation in us also, right? Like example is the preparation of the exams, right? If I am not at all making any different types of the variation during the exam preparation, what will happen? I will get the same type of the question. I will get the same type of the marks, right? How I have to vary my exam preparation? You have to follow the different types of the method while preparing the exam, right? So some of them, what you will do the mistake, guys. So you will revise the chapter. How you will do the revise? Ma'am, I have read once again the inheritance of the biology in the March, and again I will read that chapter in the April, and again I will read that chapter in the NCRT in the exam time. Is that revision, guys? No, that is not a revision. How you have to make the revision? You have to use a different type of the revision method. Once you have read the chapter, and once you have listened the chapter from the teacher, and once you have to solve the paper, and once you have to practice the questions, that is what we call it as a revision. Okay, not like simply reading. Don't do that. Practice more questions, then only you will get the revision. Yes, solve the MCQ, guys. That is very, very important. Okay. You know the topic, but you don't know how to apply the topic. Okay. So that is the main important. So then coming to the variation, I have told you that variation is absent in case of the asexual reproduction. You know that, right? In the asexual reproduction, the offsprings will be clone. We will call it as a clone, right? What do you mean by clone, guys? What do you mean by clones? The clones means offsprings which are morphologically and genetically similar, right? Morphologically and genetically similar, those we will call it as a clone, right? That is in the case of asexual reproduction. When we are talking about the sexual reproduction, variation is present, right? We are not looking like our parents, right? We are a species, correct? But are we same as our parents? No, right? So we are different than our parent. The variation is present because we are a product of sexual reproduction, right, guys? So in the sexual reproduction, it will lead to the variation. In the asexual reproduction, it is absent. Remember, guys. Okay. Yes. And the variation will lead to the evolution. Fine. Have you understood this? All the things, guys? Now you have an idea about what is a character and what is a trait. Give me a thumbs up if you understood means. Let us go fast. Okay. Tell me whether you understood or not. YouTube people, give me a thumbs up or some emojis in the YouTube. Do you understood, guys? I'm not getting any type of the response, guys. What is this? Shanta, Sneha. Tell me, guys, do you understood? Okay, clear. Fine. So now let us go to the next thing. Yeah. Some of the terms are there. I think you are familiar with this term, right? So inheritance may be either it is acquired or it may be inherited. Now you tell me, diabetes. Diabetes, it is acquired or inherited? Can you tell me, guys? In the chat box. Diabetes. Diabetes is the inherited or acquired? I'm getting only answer from Nandish. What about others, guys? Tell me in the chat box. Both. Yeah, in some cases, whether if you take about the type 1 diabetes. Type 1 diabetes, if you take means, so that is the inherited, right, guys? Type 1 diabetes is the inherited. When you're talking about the type 2 type 2 diabetes, so that is the acquired one, right? Okay. So now you tell me, I was very good in the cooking. I was very good in singing. Okay. So now you tell me that is my acquired character or my hereditary character or the inherited character, guys? Tell me. I was very good in singing. I'm not very good in singing. If I sing a song, everyone will leave the class now. Tell me. Both are only inherited or acquired? Tell me fast, guys. Acquired. Very good, Nandish. Yes, that is acquired, right? That is my skill. I have acquired that during my life, right? Yes. So that is acquired. Okay. When you are talking about the inherited, so that is comes from my parent. For example, My parent is having the brown hair. Because of that, I also got the brown hair. Some of the parents are having the white hair. The baby is also having the white hair. So those all the things are inherited. Okay, guys. So those are passed from the parents to the offsprings. So then coming to the on the what basis. So we'll get the question, right? On what basis these all the characters are transferring and how it is transferring? What is the main character was involved in it? You all know that now molecular biology, inheritance biology. Now easily you will tell that. Very good, Lakan. Yes, correct. Acquired. Right. Now easily tell that, ma'am, that is due to the DNA or gene. Right. But before the before the discovery of gene and DNA, they don't know what is that. Okay. So on the time they worked on that, the first scientist worked on that was none other than our great father of genetics, that is a Mendel. Okay. Fine. So let us talk about that, guys. Fine. So I think this is the main points in the genetics. This you should have to remember if you want to understood the, yeah. Nandish is telling if I sing the song, he will not leave the class. He will also join with me. Very good, Nandish. We will sing together. Then everyone will leave the class. Yes, Lakan. Okay. So these points are very important, guys. If you didn't understood this point, you will not able to understand what is genetics is. Okay. Let us learn about that. Okay. What do you mean by locus or loci? Okay. So it is nothing but the specific. You know what is a gene, right, guys? Either the one gene will give rise to the one protein or what? Tell me. One gene will give rise to the so many protein or only one protein. Can you tell me, for example, if you take the LAC-Z gene, okay? LAC-Z gene is a one gene, right? It will give rise to the one protein or so many protein? One protein, right? Always remember, guys. One gene will give rise to the one protein, okay? So, in that one gene, what will happen? In that one gene, okay? 1 minute, yeah. I will take some other color. Yeah. So if you take, this is the one gene, okay? This is a chromosome. Here you can see the one gene. So here that is a specific location on the gene. For example, this is a one gene, okay? The specific location on the gene, we will call it as a loci, okay? This is a gene, okay? This is a specific location. This is a specific location on the gene. So that is what we will call it as a locus or the loci, okay? It is not a gene, the position on the gene. Or for example, if you take a one bench, okay, one bench that is compared to one gene, okay, so where you will sit particularly, okay, so that we will call it as a loci, okay, guys, fine. So now let us come to the allele. For one gene, there will be a two alleles are there, okay. For example, if you take the A blood group, okay, there will be a two alleles are there, right? For the A blood group, you can take IA, okay, IA is there and the IO is there, right? There are the two alleles are there, right? Yes, IA and IO, yes. So two alleles are there, right? IA is one allele and the IO is the another allele, right? That is but one gene only, right? So for one gene, there will be a two allele, okay? For example, if you take A, capital A and the small a, right? So these are the two alleles, right, guys? So in that there will be a two things are. One is a dominant allele and another one is a recessive allele. Why we will call it as a dominant allele? It will always express, guys. Okay. Whether it will not think about whether the another person is not there, is there or nothing. Okay. So he will always express, you always dominant. Okay. That is what we will call it as. That is what we will call it as a dominant allele. Example, capital A. Capital A is a dominant allele. Okay, and small a is the recessive allele. It expressed only in absence of the dominant allele. For example, me and my brother is going in a road. Okay, so on the time I will always dominant. Okay, now I am capital A. I always dominant. I will not let him to talk. Okay, so what happened when my brother is going with his friends? Both are in the recessive manner only, right? They will talk with each other, right, guys? My brother is now dominant, right? So both of them will talk each other, right? So that is what we will call it as the recessive allele will express only in absence of the dominant allele. Okay, guys? Fine. So always remember, I fear, I fear you are a dominant. Oh, why you fear, guys? Why you fear, Nandish? Okay, I think you understood this. Let us go to the homozygous and the heterozygous. You all know that this, if the same type of the alleles are there. For example, guys, so these are the chromosomes, okay? See here, when the, in this chromosome, so here it is having the capital A and here also it is having the capital A, okay? Both are the capital A. Or if you take, here it is having the small a and here it is having the small a, okay? Those condition we will call it as homozygous, okay? For example, it is having the capital A and in this gene it is having the small a. In this allele it is having the small a. What we will call it as, guys? What we will call it as? Heterozygous. Okay, for example, this chromosome is from the father, okay, and this chromosome is from the mother. Okay, guys. Now in the mother it is having the small a and in the father it is having the capital A. So during the fertilization, what will happen? Both the things will come, no? So now the baby is having the heterozygous condition, right? For a particular character, the baby is having the heterozygous condition. So that is a capital A and small a is there, means that is heterozygous. It is a small a, capital A, or small a, both are similar. Similar it is there means homozygous. Okay, guys? Yes, yes. So same means homozygous, different means heterozygous, right? Okay. What do you mean by phenotype? Everyone know that phenotype means that is the physical appearance. Now you can see me, right? Now you can see me, right, guys? But you can't see my height, right? You can see only my face, right, guys? Yes. So which are the observable character? By looking at the person only, we can see that he is a height, he is a dwarf, like that, right? He is a short, like that, right? So that all the things are the physical character. That all we can call it as phenotypic character. Can we tell that by seeing a person that he is having the hair color that is a capital A, small a, like that, guys? No, never, right? It is not. Yes, Nandish, never, not possible, right? Yes. So, which are the character which are not visible? Okay, guys. So that is not visible. We will call it as the genetic traits. Okay, that we will call it as the genotypic. Genotypic means that is in the genetic level. Okay, guys. So that is, you always have to know that. So genotype or the genetic code is responsible for the phenotypic character, right, guys? So it is responsible for the phenotypic character. I think you understood this, guys. Can you tell me whether you understood or not? Do you understood what is a locus? Give me thumbs up if you understood, means. Tell me or give me thumbs up, guys, in the YouTube. Tell me, do you understood what is a locus? It is nothing but it is a specific location on the gene. Then coming to the allele, there are the two different types of the alleles are there. One may be a capital, we will call it as a dominant, and a recessive, we will call, sorry, it may be a small letter, we will call it as a recessive, right, guys? Then coming to the homozygous, it is having the same allele, and heterozygous, it is having the different allele. And YouTube people, I'm waiting for you. No one is answering the YouTube. What is this? Answer me in the YouTube. Okay. So then we have studied about the phenotypic character that will be observable, right? Then coming to the genotypic character that is present in the gene, right? Okay. So let us go to the next. Our Gregor Mendel has conducted the experiment. Okay. So we also have some confusion and we also get, we have to do some experiment. We have to know what is the reason for this. But we will not do that, right? But our Gregor Mendel have did this. You all know that Gregor Mendel is a monk also, right, guys? Yes. So he's having some space in his place. So he used to grow the garden pea. He selected the garden pea for his experiment and he used to grow the garden pea in that. Okay. Then he conducted the experiment. Why he selected the garden pea only, ma'am? Why he is not selected the other plant? We will tell you why he selected those all the things. You also know that, right? Let us see that. Okay. So Mendel conducted some hybridization experiment on the garden pea, and you know that he conducted that experiment for the 7 years. Guys, the statement is important. So why I am telling you the statement is important? Remember, in some exam they may give you statement wise question. In the statement wise question they may ask you the Gregor Mendel conducted the experiment for the 3 years. 3 years. Yes or no? He's conducted the experiment for 3 years. Tell me. No, he's conducted for the 7 years. Why yes? 7 years, right? Why come? How come the 3 years, guys? 7 years, right? from the 1856 to 1863. Okay. So why he chosen the pea plant? Let us see why he chosen the pea plant. So there will be a pure variety are available. Pure variety is available. What do you mean by pure variety, ma'am? I will tell you guys. What happen if I take some plant is a height in height. Okay. So if I take the height plant and if I do the self crossing, if I do the self crossing, the plant which is obtained from the self crossing will be similar like the parent. Okay, it will be a similar height like the parent. So then that will call it as a pure variety. If for example, if I take the one plant that is having the purple color flower, okay, I will do the self crossing. Okay, when I do the self crossing, so if it is a pure variety means the purple color only will come. All the progeny, if I get the four progeny means the four progeny will be the purple in color. If it is not a pure variety means what will happen? You all know that. If it is not a pure variety means one of the flower will be white color. Right guys? One of the progeny will get some different color or different height. Right guys? So whenever we do the self crossing, how the parent is there, like that only the progeny will come. That type of the variety we will call it as a pure variety. Okay guys. In the pea plant, pure variety is available. That's why he chosen that. Then coming to the pea plants are easy to cultivate, right guys? Pea plant are very easy to cultivate. No hard work. You want to do any hard work? No guys. Okay. So pea plant is easily cultivatable. So that's why he took that. And life cycle is very short. Okay. In a few months you will get the seeds. Okay. So in a few months you will get the fruits and you can get this thing. Okay. So that is the reason he chosen the pea plant. And another one, it is having the contrasting character. What do you mean by contrasting character? You know that. What is that, guys? Contrasting trait, not a character. Contrasting trait. Here we can see the purple color. Here you can see the white color. Here you can see the round seed. Here you can see the wrinkled seed. And here you can see the yellow seed color. And here you can see the green seed color. These are the contrast, right? Contrast. For example, if I wear the black color means if you wear the white color. Right, guys? If I wear the black color, you will wear the white color. That is contrasting, right? Contrast. So those type of the characters are present here. Then coming to the flower, flowers are bisexual. Bisexual means what, guys? In the single flower, both male reproductive organ, female reproductive organ are present in the single flower. Okay. Remember, in the bisexual flower, yes, Nandish, correct. In the bisexual flower, what will happen? In the single flower, both male and female reproductive organs are present. So those type we will call it as the bisexual. In the unisexual, what will happen, guys? And in the unisexual, male flower is different and the female flower is different. Okay, fine. So then coming to the flowers can be cross-pollinated only manually. Flowers can be cross-pollinated. We can do the cross-pollination by taking the anther and we only suspending that anther on the flower. Okay, so that is one thing. We can do the self-pollination, sorry, cross-pollination. You all know that what is a cross-pollination, right? You all know that what is a cross-pollination, right, guys? For example, if you take the purple color flower, okay? For the purple color flower, what I will do, I will take the anther from the white color flower and I will dust. Okay, guys? So that is what they are calling as cross-pollinating. Okay, remember. And hybrids are, yes, different plant. And when you take here, these are the hybrid plants. So hybrid plants are fertile. You know, where infertile, if you take the example, that is a mule. Mule is a hybrid between the horse and the donkey, right? That is a infertile, right, guys? That is infertile. That will not give rise to the another offsprings, right? But what happened here? The hybrids are fertile. That is only we require. That is the successful of the hybridization, right? Hybrids are fertile here. So now let us see how many characters the Mendel has chosen for this experiment. How many characters? Guys, in some exam, they were asked that how many characters they have studied. In some exam, they have asked that how many traits he studied. Don't confuse between characters and traits. In the beginning, only we have studied, right? What is the character and what is the trait? Now you tell me how many traits he studied. I need answer from the YouTube also. Tell me how many traits he studied. Traits. Answer me, YouTube student. Answer me. How many traits he studied? Fast. I need answer from everyone. Answer me, guys. 14. Very good, Shanta. Yes, Nandish. Yes, 14 traits. Okay, guys. Then coming to the characters, 7 characters. And in the exam, they may ask you which are the 7 characters. The 7 characters are very easy to remember. Height of the plant. Okay. Then flower color and flower position. Remember, guys. Flower color, flower position, height. And coming to the pod shape, pod color. Pod shape, pod color. Remember. And coming to the seed shape and the seed color. Okay. So 7 characters are there, right? See here. Stem height and the flower color and coming to the flower position. Seed color and the seed color and the seed shape. And coming to the pod color and the pod shape. Okay, guys. So these are the 7 characters. Very good, Lakan. Yes, yes, Sahul. Yes, correct. Coming to the character, he studied that 7 characters. Okay. So now let us talk about the traits. How many traits he studied? How many traits? Coming to the traits, in the height he studied the tall and dwarf. In the position he studied about the axial and the terminal. Remember, guys, so many of you will forget this. In the flower position, axial and the terminal position. Okay. Then coming to the flower color, violet and white, and inflated and constricted. Inflated means what? The hairs are filled in this. It will look like the very what we will call it as dappa dappa kanutte antivalva. What we will call it as? Here you can see. So this is a inflated, and here you can see it is a constricted. Okay. So and in the 5th color, so that is a pod shape and the pod color. So that is a green and yellow, and the round and wrinkle, and the yellow and green, that is a seed color. So these all the characters he studied. Contrasting traits, not characters, traits. Okay, fine. So now let us go to the Mendel law of the inheritance. So what he studied about the Mendel law of the inheritance. So you all know that the characters will be inherited from the parent to the offsprings. So here how he experimented this. So first he selected 2 pea plants. So that is the having the contrasting character. Either it is having the purple color flower and the white color flower. Okay. So now what he did the emasculation. What do you mean by emasculation? Student, it may ask in the step by. Okay. Steps in making the cross pollination in the pea. Remember, write down these points. First one is the selection. The second one is the emasculation. Emasculation means removal of the anther from the one plant to avoid the self-pollination. If we didn't remove the anther, means what will happen? Self-pollination will happen. And nothing is, we did the experiment there, right? So for that, we had to remove the anther. Okay. So only for removing the self-pollination. And second one, we have to do the pollination now. How? Collecting the pollen grain from the white color flower or from the purple color flower and transferring onto the female parent. So that we will call it as a dusting of the pollen grain, right? So, and fourth one is the collection and germination. So which are the plants was grown on that, we will collect the seed and we will make them to germinate. Okay, so that is what he did in this experiment. First one is the selection of the parent with the contrasting character, either the flower color or flower position, pod color or the pod shape, like that, and emasculation for doing the cross pollination and the pollination and doing the pollination and the collection of the seeds and for the germination. So now how he did that? Here you can see. So homozygous, capital T, that is having the purple, that is height, for the height, guys. Okay. So tall, and he's taken the another parent which is having the pure breed, that is a dwarf, small t and the small t. So now what happened in the F1 generation? All the offerings is having the capital T and the small t. Okay. So capital T and the small t. What do you mean by that, guys? So here it is having the heterozygous condition. Okay. So what happened? In the F1 generation, they have did the self crossing. Okay. Selfing. So in the only one generation, they are doing the cross pollination. In the second generation, it is the selfing only. They are not removing the pollen grain or nothing. Just selfing is happening. So in that, we have got the different types of the plant. So in the different plant, height of the plant. So in the F2 generation, we have got that 3:1, right, guys? So that is 3:1. Okay. So parent, so that is the 3 is height one and 1 is the dwarf one. Okay. So now let us talk about the inheritance of the one gene. So inheritance of the one gene means, so here we are concentrating only on the one character, that is the height of the plant. Okay, guys. So in the inheritance of the one gene, the phenotypic ratio is 3:1. Remember. So here the cross between the height plant. So capital T and the small t and the small t. We have got F1 generation, that is the capital T and the small t. Okay, guys. So we have got the phenotypic ratio 3:1. 3 plants are height, 1 plant is dwarf. Right, guys? Then what is the genotypic ratio? Phenotypic ratio 3:1. What is the genotypic ratio? That is different. So that is 1:2:1. Always remember, guys. Okay. In the Mendelian, we have got the F2 generation. That is the genotypic ratio 1:2:1. So then coming to the test cross. Why we will do the test cross? We have got F1 progeny. We don't know whether that is having the homozygous or the heterozygous. Right. So how to find out that whether the F1 progeny was the homozygous or heterozygous? We have to do the crossing, right? So that we will call it as a test cross. How we will do the test cross? We will take the one of the plant which is obtained from the F1 generation. Then I will do the cross pollination with the parental, which is having the dwarf. Okay, so that is having the recessive parent. I will cross with the recessive parent, guys. Okay, so remember always the test cross will do with the unknown genotype, and with the recessive parent only. Remember, unknown genotype and with the recessive parent only. Okay, guys. So now what will happen? So if the F1 generation that is having the heterozygous mean, so we will get the ratio that is 1:1. In the test cross, what we will get? 50% height plant, whereas the 50% will be dwarf plant. That is the 1:1 ratio. Remember, guys. So that is 1:1 ratio. Okay. So here you remember it is used to determine whether the F1, the individual obtained from the F1 is homozygous or heterozygous. Always remember, guys. If you want to know what is the genotype of the F1 generation, I will do the test cross. Okay, fine. So let us go to the back cross. What is the back cross? Back cross means what? So here the F1 generation was obtained, right? I can cross that with the any parent. Okay, guys. Either I can cross with the capital T. If I do the capital T crossing means I will get all the progeny will be tall only. That is one only. Okay. All the progeny will be tall only. Okay. When I did the cross pollination with the dwarf or the recessive parent, I will get a 1:1 ratio. Okay. Back cross will be with the both the parent. Backing means stepping towards the back. Stepping towards the back means what? The parents are there, right? So I can cross the F1 progeny with the any of the parent. This usually done in the hybridization process. During the hybridization in the animals, they will use to do to know the genotype of the plant. Genotype of the animals. It is useful in the genetic studies for isolating the certain characteristics. So related group of the animals and the plants. Always remember. So it is due for the finding the certain characters in the animals. Usually in the animals they will do. Okay, guys. Fine. So that is what we will call it as a back crossing. Okay. So here you can see with the dominant parent also did, with the recessive parent also did. But test cross is only with the recessive parent. Okay. So why we are doing that? To know the genotype of the F1 progeny. Fine. Are you understanding, guys? Samuel, Lakhan, Sahul, I am going little fast, but are you understanding, guys? Samuel, Lakhan, Sahul, Zoom people, tell me, guys. Nandish, Nagveni, Shanta, tell me, Pa. Is I am going very fast? Are you understanding? Okay. So let us now understood about the dihybrid process. Always remember, guys, when we are talking about the dihybrid, monohybrid means what? Single character. Dihybrid means what? Two characters. Here I am taking the two characters. And what you have to remember here, there the ratio, phenotypic ratio you have got is 3:1. So here what you will get? So here you will get the phenotypic ratio, that is 9:3:3:1. So that is phenotypic character. Okay. Then coming to the genotype. Genotype is very easy to remember, guys. Okay. How you have to remember? 1:2, 2:1. That we have seen in the monohybrid cross also, right? Here you can see 2:4:2. Okay. Then again 1:2, 2:1. From the starting also 1:2, 2:1. From the last also 1:2, 2:1. In the middle 2:4:2. That's it. You can remember, right, guys, easily. 1:1 in the starting, 1:1 in the last also. But in the middle 2:4:2. That's it. That is a genotypic character. Genotypic things. Okay. Genotypic ratio. Coming to the phenotypic ratio, that is 9:3:1. So in the dihybrid class, I am taking the 2 character. So what are the 2 characters here Mendel took? So that is 2 character is the one is a seed. Seed color is the yellow color and another seed color is the green color. And yellow color seed is having the round shape and the green color seed is having the wrinkle shape. Okay. So he's taken the 2, right? One is the color and another one is the shape. So dihybrid, guys. Okay. One is he's taking the color. Always remember, guys, color. And the second color is the shape. And the second trait is the shape. Okay. Character. Character is the shape. Let us learn about that. How we did. See here. Always remember, here in the yellow color round seed, we are having the 2 genes. Right, guys? 2 genes. Remember, one gene is for the round and one gene is for the shape and another gene is for the color. Okay. So R is for the color, yellow color. Okay, guys, yellow color, right? Yellow color. And Y is for the what? And Y is a dominant. Y is dominant. That will show that it is a round. Right, guys? If it is a capital Y is there, means that will tell that is a round. Okay. And coming to the small r, if the small r there is means, so that will show that that is having the green color. Green. R means color. Then coming to the Y, small y will tell that that is a shape, that is a wrinkle. Wrinkle. And how I have to take the genotype? How I am taking the genotype? Capital R and the capital Y. Okay, guys. See here. From here I am taking the capital R and capital Y. From here I am taking the small r and small y. So how can I give the heterozygous in the F1 generation? See here. R1. So see here. Capital R, small r and the capital Y, small y. Right, guys. Capital R, small r. Capital Y, small y. First you have to write the R. So then you have to write the Y. Okay, guys. See here. So now I am doing the selfing between the F1 generation. You know what is the F, right, guys? You know what is the F, right? F means filial. 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 Okay, guys. Fine. Lakan, in the test cross, there is a no other examples. You can do, for example, for the test cross, you can take the others also. For example, you can take the round seed and the green seed. Okay. So if you take only concentrating on the only one color, so round seed and the green seed means you can take the capital R and the, so in the F1 generation, you have got the capital R and the small r, right? Capital R and the small r, 1 minute. So here I will explain. So if you take capital, the F1 generation, you have got capital R and small r. Capital R means it will tell us about the round, sorry, yellow, right? And the smaller will tell us that that is a green color, right? So what will happen now? Capital R, smaller is the, that is the F1 generation. Now I will cross that with the smaller and the smaller because I don't know what is the, whether it is the heterozygous or the homozygous. What I will do now? Capital R, see here. So, and the smaller. Here it is having the only one, right? So now I will cross that. Capital R smaller, I will get the capital R smaller, smaller. So I have got 2 things, right? One is yellow color and another one is a green color, right? So means it is a heterozygous, right? Right, Lakan? Now you understood, Lakan? We can take any examples, Lakan. Can't we take the HEPA hybrid? What do you mean by HEPA hybrid, pa? Hepta hybrid. So we can take hepta hybrid pa, but that is difficult to know. It is very difficult, right? Knowing all the things, hepta hybrid. We can take 7 traits together, but that is very difficult to study. You can't study. Yeah, you can try. You can try, but no one tried that. You can try, but that will be very difficult, guys. Studying those all the character, and it may be not. You can't study also because they are independent, right? You can't study also that all the things. Maximum you can study 3. Yeah, us. Yeah, we all will do that. Lakan, Samuel, Nandi, Shanta, and others who are here. Yeah, Nagveni, we all do that. Okay. Yeah, I will also join with you. Okay, guys. Yeah. 1 minute. Yes, guys. So now you have to take the hybrid. So in the X2 generation, how we are going to take? So see here. So here you have to take. Remember here, guys. So capital R and the capital Y. Capital R and the small y. Okay. And you can take small r, capital Y and the small r, small y. So now we have got the 4 gametes, right, guys? 4 gametes. So now we have got the 4 gametes. We have to do the crossing now. See here. Capital R, small, capital R, small, capital Y, capital R, small y, and the smaller capital Y and the smaller and the small y. So like this you have to take. So here also and here also, male and the female. When we cross both of the things, we can do this by the Punnett square. Okay. If it is a more or there means we can do it by the Punnett square. This we will call it as a Punnett square. Punnett square. Okay, guys. So here we have got the 16. 16 offsprings we have got. In the 16 offspring, so that is 9 is having the round and yellow, and the 3 is to 3 is to 1. So and 9 and 3 is having the round and green, and 3 is having the wrinkled and yellow, and 1 is having the wrinkled and green. Now you tell me, guys, by observing this, what you came to know? Now you tell me, guys, by observing this, what you came to know? Either it is undergoing the independent assortment or non-independent assortment. Tell me, guys. Yes, very good, very good. Tell me, guys. 2 genes are there. Whether 2 genes are assorting independently or together. If it is assorting together means we should not get any type of the hybrids here, right? Right. Both the genes are assorting independently here. Okay. So Lakan, are you understanding, Pa? Samuel and others? Because of no intermediates. Yes. Yes. So now you can tell me, you can see here. So that is having the 9 is to 3 is to 1. But if you take particular gene, guys, if you take particular gene, so that is a yellow color. Yellow color. If you take the particular character, so that is a color that is assorting independently, right? That is assorting independently. That is giving the 3 is to 1 ratio. Whereas another gene, if you take means that is also assorting independently. That is also giving 3 is to 1 ratio. If you combine that, so square that, it is getting the 9 is to 3 is to 3 is to 1. Right, guys? Yes. So then we have got the phenotypic, that is the phenotypic ratio. We will get the genotypic ratio. 1 is to 2 is to 1, and 2 is to 4 is to 2, and 1 is to 2 is to 1. Right, guys? Very good, Lakhan. Very good, Samuel. Yes. So who is not understanding? Nandish, what you didn't understood? Loss. So nothing. So guys, if you take these 2 characters are assorting independently, right? If you take the particular one character, that is the color of the seed. If you take the color of the seed, it is assorting independently, right, guys? If you take the only color, how I explained here, that is the in the test cross I have explained, right? When the Lakan asked me, on the time I have told you that. So here you can see that I have told you that if I take the capital R and the small r, capital R is the yellow color, small r is the green color, right? So then what is the progeny I have got in the F2 generation? That is 3 is to 1, right, guys? So that is obviously it is independently assorting, right? So when you talk about the shape also, that is also assorting like in the 3:1 ratio, right? So that is what I have told you, right? Yes, law of the segregation. Yes. So now let us go to the next. Yes. So dihybrid cross, we have studied that. I have told you that phenotypic ratio also. Now let us talk about the law of dominance. When the two characters, when the two alleles are together, okay, the characters are controlled by the factors. We know that factors are always together in the pair, right? One is the dominant and another one is the recessive, right, guys? So we always know that when the dominant and the recessive is there, always the dominant one is going to be expressed. The recessive one is not going to be expressed, right? So that is what we will call it as law of dominance. That is the first law of Mendel. Okay. So here you can see. So capital T, small t is there. Okay. So capital T, that is having a similar pair of the character. So small t, that is also having a similar pair. When it is having the capital T, small t, here small t also there, capital T also there, but expressing is a tall, right? But the dwarf trait was not expressing, right, guys? So that is what it will tell, that dominance, law of the dominance, okay? It will inhibit the expression of the small t. Okay, guys, fine. So when crossing the two homozygous parent, the resulting offspring is a uniform. That is called as a uniformity. That is not important. Let us go to the law of segregation. So that is the second law of the Mendel. Always remember, guys, a law of the segregation. We also call it as law of the purity or law of the purity of the gametes. So let us see what is that. During the gamete formation, what will happen, guys? The gamete will segregate independently. Okay. For example, capital T together is there. Okay. During the gamete formation, what will happen? You know how to write the gametes of the T. Capital T, capital T. Can you tell me how can you write the gametes of the capital T and capital T? Tell me, guys. Or you tell me how will you write the gametes of the heterozygote? Tell me, guys, how you will write the gametes of heterozygote? Capital T and the capital T. Together you will write or separately? Give the space. If you are writing means give the space. Capital T separate gamete and the small t is a separate gamete. What happened here? Are they coming together, guys? No, right. They are segregating, right? For example, if you see, whenever the exam was nearer, what the first thing you will do? Tell me, guys. When the exam is nearer, I will tell you what the first thing you will do. Sleep. Not everyone will sleep. What first you will do? When the exam timetable came, you will do that. First, you will separate the physics books. Second, you will separate the chemistry books and biology books. Right, guys? You will do or not? Tell me. I will do that. You will segregate the books or not? Tell me. Pakka you will do that. Tell me, guys, whether you will do it or not. People from the YouTube, tell me, you will segregate the books or not? Oh, you will not segregate. Shanta will do. Shanta and me is same. Yes, Shanta, even I will also segregate the books. Otherwise it will create a confusion, right? Very good, Samuel. Correct, Pa. It will create the confusion, right? Yes, we have to segregate. Then I can easily, when I'm studying the physics, I can go to the easily physics books and I can study whatever the needs I want, right? Yes. See here, guys. Nandesh, too bad. Whenever you are studying, segregate the books. Okay, Nandesh? Okay, fine. So let us see here what will happen during the gamete formation. Okay, guys, during the gamete formation, what will happen? The factors are the allele. So you know that the allele is nothing but the factors, right, guys? So allele of the character, for example, capital A, small a is there. During the gamete formation, capital A will separate and the small a will separate. Okay, that is only we will call it as segregation, gamete segregation. So together the gametes are there, right? Together the alleles are there. What happening during the gamete formation? They will segregate. We will get, for example, if you get the diploid. Diploid is there means during the meiosis, what will happen, guys? It will become a haploid, right? It will become the haploid. So that is what they are telling here. Okay. So it was present in a parent from the each other, such as a gamete. So recessive only one of the two factors. Okay. So for example, homozygous parents produces a similar gamete. For example, if you take female is having the XX. Okay. The male is having the XY. Okay. So the male will produce two types of the gamete, right guys? X and the Y. They are segregated. Here X and the X. They are segregated. And during the fertilization, what happened? If this it is having the X, it was the gamete was fused with the another gamete which is having the Y. Okay, what will happen now? Fertilization will happen. Now again the diploid stage was restored. Right, guys? So this is what we will call it as the segregation, law of segregation. Okay, guys. So here the allele will segregate during the meiosis. Okay, during the gamete formation. You all know that how the gamete will form by the meiosis, right? Yes. And the heterozygous parent produces two kinds of the gamete. I have told you one allele in the equal proportion. Yes. Yes, Samuel, correct. Samuel also will separate. Nandish, only you are not separating. Everyone is separating the books. Okay, guys, that's it of the law of segregation. So if you understood what is the law of segregation, it is very easy to remember the in the chromosome. Chromosome also, in the chromosomes also, chromosomal theory of the inheritance. There also it will same thing will happen. Okay, now let us come to the law of independent assortment. The same thing happened, guys. During the dihybrid cross, we have studied that. So here, the one character is not dependent on the another character. One gene is not dependent on the another gene, right? So they are segregating. For example, what will happen? Here, the one gene is there. Okay. So, for example, guys, here the one gene is there. Okay. So here, the gene which is responsible for the color, the gene which is responsible for the color, and the gene which is responsible for the shape, both are together. Okay, both are together means what will happen? They will independent. So, for example, they are not together. I am telling together. Sorry, that is in the linked gene. Okay, 1 minute. So they are not together. Okay, for example, see here, they are not together. One gene is here and one is gene is here. It is responsible for the color and it is responsible for the shape. Okay. So whenever they are going, whenever during the crossing recombination, they will go separately. They will adsorb independently. I will give you an example here. For example, you and your friend guys. Okay. So now you are in Karnataka. Okay. You are in Karnataka or you are in some place. You all are in some place. I don't know, guys, where you all are there actually. I'm taking the example of Karnataka. I'm in Karnataka. Okay. My friend is in Andhra Pradesh. Okay. My friend is in Andhra Pradesh. Can we go together everywhere? I want to go for the shopping now. I want to go for the shopping now. Can we go for the shopping now together, guys? Is it possible? If we want to go for the shopping together means, how come it is possible? Not possible. Right, guys? So that's why what we will do. If I want to go for the shopping, I will go for the shopping. If she want to go for the shopping in Andhra Pradesh, independently she go for the shopping. Right or not? Tell me, can we go together, guys? Not possible. Oh, online shopping or video, it is possible. Together, can we go together physically? Physically, I'm telling. Physically, not possible, right? Yes. On the time, what will happen, guys? Remember always, when the genes are separate, the distance is very more. Okay. So when the distance are very large, on the time, what they will do? They will independently adsorb. Independently adsorb. Okay. So that is the same thing happening here. So the color is in the different gene, whereas color is in the different gene, whereas the shape in the different gene. So they will adsorb independently. Okay, guys. That is only here telling Mendel. Mendel law of the independent adsorbment states that the allele of the different gene adsorb independently of one another during the gamete formation. Remember always, when these two genes are distance are very large, when the distance are very large, on that time only independent assortment is possible, guys. Okay. So when is distance is large, distance is large. For example, see, me and my friend is in the same apartment. Now you tell me, guys, me and my friend is very close. Okay, we are very close. Wherever we go together only. Now you tell me, we are in the same apartment. Can we go together? Can we go together for the shopping now? Tell me, possible or not? When we are in the similar apartment, can we go together, guys? No, Ante, no. Why it is not no? Why it is not no, Nandish? We are very close, Nandish. Possible. Very good, Nagveni. Nagveni know that. Yes. When me and Nagveni were in the similar apartment, Nagveni and me will go for the shopping together only. Don't want to shop. We want to do the shopping. On that time, we will go together. Remember, guys, what will happen here? When the genes are together, means the distance is very less. Okay. When the distance is very less, we will call it as a linked gene. Okay. Linked gene. So here only you have to remember that in the linked gene, the law of the independent assortment will not work. Okay, guys. In the linked gene, the law of independent assortment will not work. They will assort together. They will not assort independently. Okay, remember here, law of independent will not work for. Law of independent assortment. Independent assortment will not work for will not work for linked gene. Linked gene. This is asked in some exam. In the statement wise question, they have asked this question. Whether the law they have given the statement like this, guys. In the law, so in the linked genes, it will show the law of independent assortment. Lakan, that only I am telling. Linked gene means what? So here the color for the character for the color, the gene which is responsible for the color and the gene which is responsible for the shape, they are together. Means the distance is very less. Okay, where the distance is very less, that we will call it as a linked gene. Do you understood, Lakan? These are linked gene I am telling, right? See here, this is for the color. This is for the color, seed color, and this is for the shape, seed shape. They are together. The distance is very less. Distance is very less, Lakan. On that time, what will happen? These type of the genes, we will call it as a linked gene. Linked gene. So during the recombination, what will happen? This both the things will go together. This both the thing will go together. Okay. For example, this is having the seed. The seed is having the round color. Sorry, it is having the round shape and the yellow color. Yellow color, the shape is round and the yellow color, it is linked. Linked means what will happen? All the progeny will come with the round and the yellow color only. We will not get the round and the green color. Round and green color is not possible. Okay. So, because these are linked, which are the round seed is having, it must be yellow in color. Which are the green seed having, it is a wrinkle in shape. Those things we will call it as a linked. Okay. So Lakan, do you understood, Pa? In the linked gene, what happened? The distance is very less. So that's why wherever they are going, they will go together. Do you understood, Lakan? Lakan, tell me whether you understood or not, Pa? Guys, do you understood? Who all are here? Do you understood, guys? Shanta, Nagveni, Nandish? Always remember, this is the important point. The linked gene will not show the law of independent assortment. Okay, fine. So I think you understood. So the inheritance pattern of the one trait will not affect the inheritance pattern of the another. Okay, so inheritance pattern of the one gene will not affect the inheritance pattern of the another. We will call it as the law of independent assortment. Independently they are going. Okay, fine. For example, if you take another example, for the law of independent assortment, me and my sister is in the similar room. Okay. So whenever we are going outside, she will go independently. She will go alone and I will go alone. Right, guys? That is what we will call it as an independent assortment. Okay. When me and my friends are together, what we will do? Whenever we are going together, whenever we want to go outside, we will go together. That is what we will call it as dependent assortment. Okay. Fine, because we are linked, right? We will not go independently. Fine. So non-Mendelian inheritance. These are very, very important. Non-Mendelian inheritance. Always remember, guys. Always remember. Non-Mendelian inheritance. It will give F1. In the F1, what will happen? There we have studied that phenotypic will be 3-1 in the Mendelian. Remember, in the Mendelian, we have studied that 3:1, right? 3:1, that is the F1 generation. In the F2 that is the phenotypic, right? Phenotypic we have got is 3:1. And the genotypic we have got is the 1:2:1. Right, guys? In the genotypic. But what happened in the incomplete dominance, codominance, and the multiple allelism, the genotype will be similar. Genotype and the phenotype will be similar. That is 1:2:1. Okay, 1:2:1, guys. All phenotype also same and the genotype also same. Remember, guys, they may ask you what is the ratio of the F2 generation in the codominance? Nothing but 1:2:1. What is the phenotypic ratio? 1:2:1. What is the genotypic ratio? 1:2:1. Similar, guys. Similar. In the incomplete dominance also similar. 1:2:1. In the multiple allelism also same. Similar. Yes. We will study about this examples and everything. Okay. So which are the non-Mendelian inheritance? Always remember. Incomplete dominance. What happened in the incomplete dominance, guys? What happened in the incomplete dominance? For example, you can take the both the colors are there. White color flower is there, and the red color flower is there. Okay, so the red color flower actually dominant, right? But it will not completely dominate the white color. So what will happen now? Both will express in the F1 generation. They will fight. So you're also dominant, I'm also dominant. I will not let you to dominant. Okay, so what will happen? So here the F1 is so that is red color is dominant, but that is not completely dominant on the white color. So that's why in the F2 generation, what will happen, guys? In the incomplete dominance, we'll get a pink color flower. Pink color flower we will get, where white also expressing, whereas the red color is also expressing. We will get pink color flower. Okay, guys, what is the ratio of the phenotypic character? That is 1:2:1 only. What is the genotypic ratio? 1:2:1 only. Right, guys? That is incomplete dominance. Always some of them will confuse between the incomplete dominance and the codominance. Remember, guys. So that is in the Mirabilis jalapa and the snapdragon. Mirabilis jalapa and the snapdragon are example for the Mirabilis jalapa. We also call it the 4 o'clock plant, right? Yes, Lakan, I will end the class now. Okay. So the example is if you are getting the pink color flower, that is the incomplete dominance. Okay, guys. The other non-Mendelian characters are codominance. Other one is the multiple allelism and the polygenic inheritance and the pleiotropy. Okay, guys. So always remember, these all are the non-Mendelian characters, non-Mendelian inheritance. They may ask you in the exam. That is like an exam. Give the example for the non-Mendelian inheritance like that. Okay. So remember these all the things. Don't miss the next class. Okay, guys. All are watching this. I will tell you, don't miss the next class. We are doing very, very important topics in the next class. Okay, guys. Thank you so much. Let us continue in the tomorrow's class. Okay, guys. Bye. Nandish, what you have to do is you have to solve the more questions. Okay. So, Nandish,